Never fear, my friends. I have shown up. Some of you worried. I know. What do we got? 20 people today? That's not so bad. So I wanted to warn you. Someone, Nishi, asked me, where is the recording from last Friday? Well, unfortunately, with Zoom, if you don't save one recording, you can't make another one. And I had done all these advisement meetings, and I forgot to I forgot to save the recording after my last advisement meeting. So then when I tried to record the lecture, it didn't record. So I don't have a recording of it. But we're starting to overlap with what I did last year. So I have the exact same lecture in my YouTube channel. I've got my own YouTube channel. I've got 22 subscribers. Talk about monetizing. So if you want to see that lecture, let me know, and maybe I'll just post that in Moodle because it should be there anyway. So it's Yusuf. Hello, my friend. Good to see you again. You looks like you're outside.
Yeah, I'm outside right now.
Yeah, I was outside all morning. It's getting cooler out. So the clouds came and hid the sun.
Yeah, it's very nice out today.
Okay. All my favorite students showed up. This is great. The losers didn't come today. Losers. Uh-oh. Okay, so if you didn't come today and you're just watching the recording, I'm sorry for calling you losers. I don't mean it. Okay, so go ahead and do the quiz. Quiz just opened at 1:03. It will close at 1:13. So you've got 10 minutes, only one question, and I hope you do well. Later, dudes. I still have my audio on, so if you want to talk to me, I can talk back to you. Okay, well, I think that closed. So let's take a look at it if we can. Let me see if I can find it first. Bum, ba, bum, 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 ba, bum. Bum, bum. Bum, 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 bum. Okay. Okay, here we are. So I think this is pretty easy. I mean, if you knew your notes, you knew where this was, it wasn't so hard. So signaling involved neurocrest induction dependent on, oh boy, mine are in order, so this is going to be really easy. FGF and Wnt, which induces neural plate from the ectoderm by upregulating SOX2. The width of the neural plate is limited by a BMP4 gradient that comes from the lateral mesoderm. In the area between the neural and non-neural ectoderm, where this hormone is at its lowest levels, the neuroectoderm becomes neural crest cells by upregulating SOX9 and 10, which makes these neural crests. This upregulates SNAIL, which causes epidermal to mesenchymal transition, and then more BMP causes slug and twist to come up. Et voila. That's French. I just slipped into French there. It means there it is. Okay, people, I hope that was as easy for you as I hoped it was when I made it. Okay. Okay, so today, and by when I say today, I mean to like this day we're in right now. Today, we will be going into chapter 7. Okay, I'm just reestablishing my real estate here. Okay, so chapter 7. We're not going to do all of chapter 7, just parts of it. It's kind of an interesting chapter. The concept of it is kind of neat. It's about how this doesn't have anything or not much to do with signaling or signal transduction. It has more to do with how cells attach to each other and how that attachment it activates signal transduction in the cells, but also allows cells to kind of pull on each other and then change the shape of the tissue. So this is kind of an example of it here, although we don't do this in class, but I thought this was really cool. I dug it up on the internet somewhere. You can click on it to find out where. But this is about the development of the heart. And here you've got mesodermal cells that become blood vessels, basically. Here you've got 2 blood vessels, and these 2 touch each other and then fuse together. So this is not what I'm talking about yet. So here's what I'm talking about. Once they fuse together, surface tension on different parts are squeezing here, but not here and here. And the whole thing twists based on this surface tension. And that's what morphogenesis is, the genesis of the shape of the thing, which is often due to this kind of surface tension squeezing layers of cells into a certain direction. We'll see this again. Well, we saw this before with gastrulation in the xenopus, how gastrulation in chick and mouse, it was all about mesenchymal cells letting go of each other and crawling through the node and going forward. In xenopus, it was a sheet of cells pulling itself along. Okay, and that's because the mesodermal and endodermal cells were attached to each other, but they weren't really attached to the ectoderm. They were just crawling along it. So if you think about this layer of mesoderm, they're really tightly attached to each other. So as they move, they pull each other along. So that's the surface tension thing I'm talking about. And also, we'll talk later in this chapter about how neurulation, how that you have a neural plate that then squeezes up and becomes a neural tube. So that's what we're talking about here, how that shape change happens. So anyway, as I said moments ago, it has to do with cell-cell connections. So what are these cell-cell connections? I'm just checking my notes to make sure I haven't forgotten something. So we've got two, but really three different types of cell-cell connections. I guess three different types of cell-cell connections. One are adherens junctions, which we talked about back in chapter 5 when we were talking about hippo signaling. If you remember, hippo is attached to the adherens junctions unless the cell was connected all around its surface. So we'll talk more about that here. But those were adherens junctions. And adherens junctions are about the protein cadherin. So there are 30 different types of cadherins. And cadherins are transmembrane proteins. Here you can see you've got an external domain, transmembrane domain, and then an internal domain. And these are connecting cells to cells. And they reach out and they grab the same cadherin from the opposite cell. So this is called homophilic. So they're like cadherin E will only attach to cadherin E. It won't attach to other cadherins in this type of junction. So this is a calcium dependent process. So you need calcium on the outside here to get them to attach to each other. And at the same time, let me go to the next slide. Oh, so these cadherins then are used in two different types of junctions, the adherens junctions and the desmosomes. So I got a little ahead of myself here. This is the adherens junction. So the adherens junction is the homophilic one where cadherin E attaches to cadherin E and they attach to each other. And these are attaching to the cadherin of the opposite cell on the outside. On the inside, they're attaching to the cytoskeleton. And in terms of adherens junctions, it's microfilaments of the cytoskeleton. So, you know, I was trying to think of a metaphor for what would make sense. What would make this make sense? So you got to figure cells are not like, it's not like us. Like if I'm not touching another human being, I'm still attached to the earth, right? Gravity's keeping me down. I can move based on the friction between my feet and the floor. I can walk around all I want. If I was up in space, right, I'd be floating around. And if there wasn't something to grab or push off, I'd just be floating around, right? So imagine I'm up there floating around in space and I grab a handle, like some kind of like a dog chew toy that's got like two handles attached to each other. Okay, that's my cadherin, okay? And then someone else comes along and grabs the other side of it. Now I can pull on that other person can pull on me. That's kind of what these things are doing. You're reaching out from cell to cell, so you're attaching to the other person, but you're also attaching to your own skeleton here. So cadherins here. And again, it's a calcium dependent attachment between the two different cells. And the transmembrane domain is here. And then here's where we're attaching to the microfilaments. Let me blow this up so we can see it better. At least so I can see it better. So this, if you're in cell biology right now, I'm going to be repeating a lot of what I said in cell biology. I probably said this before in my class, but I think you should have cell biology before this class, which makes no sense because this is a 300 level class and that's a 400 level class. But you know, all the stuff I talk about in cell biology, in this class, we see application of it. So that's what I really like about this class. Excuse me. But these you can think of as stress fibers, which I'll talk about again later in a minute. So you've got anti-parallel arrays here of microfilaments, and they're attached to each other with alpha-actinin, which is just a bundling protein. And then the cadherin is attached to the microfilament by a whole bunch of proteins, okay? But we're only looking here at alpha and beta catenin. Remember beta catenin, Wnt pathway, same protein. Here it's usually being used as a structural protein, which is kind of weird. You expect to see it as a transcription factor, signaling molecule, but here it is attached to the site here it is being used to attach cadherins to the cytoskeleton. And then here's some other proteins. Don't worry about them. We're not going to memorize them. If you remember this bad boy from cell biology, ARP23, that was involved in branching of microtubules, which we see in the cell cortex. So little foreshadowing there. Okay, so we've got our cadherin then that you get a whole bunch of them in one spot, like a little spot weld of a bunch of cadherins. That's an adherens junction, and it grabs to an adherens junction on another cell. And signal transduction goes both ways, which is really important. So one important aspect is when this cell grabs another cell, then it knows it's grabbed another cell because that activates signal transduction caused by some of the proteins that are attaching this thing to the cytoskeleton. So that'll activate some kinases, and that alerts the cell that it's attached to another cell. And signal transduction can go in the other direction as well. So when the cell undergoes, EMT transition, when it goes from epithelial cell, where they're all attached by these cadherin junctions, into mesenchymal cells, that is relayed by some signal transduction inside this cell that tells it to let go of the cadherins of the other cell. So it's both structural because it's holding things together, but it's also signaling going on here. Okay, so that's our adherens junction. Then we've got our desmosomes, which are very similar. The big difference here, two big differences here. One is that they are heterophilic, which means you've got one type of cadherin here attaching to a different kind of cadherin there. So they're not the same. And then also they're attaching to intermediary filaments. So we'll talk in a second about cytoskeleton some more. But intermediary filaments are kind of tissue-specific structural proteins that they're not involved in contraction or anything, but they will kind of like when the cell stretches, they'll stretch and allow it to snap back against it. And they're more structure inside the cell. So it's just attached to a different kind of structural protein. And these are attached here by gamma and beta catenin. So that's what attaches the cadherins to the cytoskeleton in these desmosomes. So, okay, here's desmosomes again. Here's your cadherins, and you can see you've got red ones and yellow ones, which just signifies two different cadherins. And then here's our beta catenin and gamma catenin. That's attaching it through this desmoplakin to an intermediary filament. Don't worry about desmoplakin. You're probably going to laugh at me when I say this, but I try and decrease the number of things you need to memorize to a bare minimum. So don't bother memorizing this. Actually, if you look at this, oops, sorry. Oh, I can't do it here. But there's actually two more words underneath this, so you can memorize those two words too if you want, but they won't be on an exam. Okay, so that's our cadherins. The other type of connection, well, the other protein involved in connections is the integrin. So integrins then are going to attach to extracellular matrix. So extracellular matrix is everywhere, as you know. So if a cell is going to let go of cadherins of the cadherins of its same cell and then wants to crawl, it needs some way to crawl. And that's what these integrins are doing. They're allowing it to crawl across extracellular matrix without touching other cells or without grabbing onto other cells anyway. So our integrins then are heterodimers, alpha and beta subunit. And actually the alpha is two separate proteins attached by a disulfide bond. That's not that important. But this extracellular domain will attach to fibronectin, collagen, and probably some other proteoglycans and glycoproteins of the extracellular matrix. On the inside, then, you've got binding of signal transduction molecules again, and adapter proteins. So we can see that here, these integrins all come together into a focal adhesion. So here is the extracellular matrix, fibronectin and collagen attached to. And then on the inside, you've got a protein talin and vinculin, and that attaches it to this microfilament. So, and then here's alpha-actinin again. So we saw that momentarily ago. That's a terrible way of saying that, Dr. Slish. Here's alpha-actinin attaching microfilaments together. So that's just kind of the same thing here. It's a bundling protein keeping microfilaments attached to each other. So we've got these proteins then attaching our integrins to the cytoskeleton. And let's hang on, let me go ahead a little bit. Nope. Okay. In addition to these are kind of like adapter proteins that are just attaching one protein to another for structure. But in addition to that, you've got a whole lot of other proteins attached here that you don't see. If you take cell biology of cancer, we'll talk about some of those. And they're involved in signal transduction again. So again, it's like when the integrins are attached to this extracellular matrix, that signal, that instigates a signal that goes into the cell to let the cell know that it's attached to something. And people found out this the hard way when they first tried to put cells in culture, because they take a tissue, like say, take a chunk of liver, hit it with collagenase, and collagenase will break all this collagen down. And now you've got free cells. And you take those cells, you put them into culture, and they die. Like immediately, they just die. And why is that? Well, then they found that if they put collagen on the bottom of the dish and then put their cells on, then they'd be okay. Because it turns out that these integrins, when they attach to this extracellular matrix, they signal get signal transduction happening on the inside, and that tells the cell, look, you're attached, you're going to be okay, just stay alive. Stay alive, I will find you. That's a quote from an old movie, Last of the Mohicans. Ignore that quote. So anyway, in cell biology, if you're in that class, we'll talk about this probably next week. It's about trophic factors. It's keeping the cell alive so it doesn't go into apoptosis. And that's kind of a protective mechanism against cancer, because your most dangerous cancer cells are the metastatic ones, the ones that leave and go into the bloodstream. Well, most cells, when they leave attachment, will just die because their integrins won't be attached to anything, and so they go into apoptosis. So that's the signaling from the outside to the inside. On the inside too, you've got signaling that tells it whether it should attach or let go. And that's important for cell crawling, which we'll talk about in a minute. But the integrins grab and the cell pulls itself forward, and then they let go, they go forward and they grab somewhere else and they pull themselves forward again. So signaling, again, like the cadherins, is going in both directions through these focal adhesions. So we've got 3 different types of spot welds here. We've got cadherins in adherens junctions, cadherins in desmosomes, and integrins in focal adhesions. And this is keeping cells in a tissue. Okay, so cytoskeletal elements. So again, this is kind of review of cell biology. So if you've had that class, this will help you for your final, because you will see that again in a few weeks. So we've got 3 different types of proteins involved in cytoskeleton. You've got your microtubules, your microfilaments, and your intermediary filaments. Both the microfilaments and the microtubules will be involved in movement, either of the whole cell or things inside the cell. And your intermediary filaments will not. They're just structural. So, but they will kind of like stretch and respond to things. So they're giving the cell integrity in that respect. Let me check my notes real quick here to see if I have forgotten something. Okay, so let me just say something real quick, which I didn't talk about, but I said it quickly, but um, one of the important things about these adherens junctions is that this is what's keeping your cells attached to each other in a tissue, right? And when that tissue, when the sheet of cells starts to move, it keeps them all together and it makes them one coherent thing. When cells undergo that epithelial to mesenchymal transition, they let go of those cadherens junctions, and then they can move away. And when I talk about epithelial tissue, it's different here in embryos versus in adults. Epithelial cells we think of as skin, right? Your outer layer, and between that outer layer of skin and the dermis, between the epidermis and the dermis, there's this really thick layer of protein there that provides protection, so things don't come in through that way, and it also keeps cancer cells from getting into your body and killing you, because most cancer cells are epithelial in nature. So, but here in embryos, epithelial cells aren't necessarily skin cells. They can become skin cells, but they can become a lot of other things. They're just a sheet of cells that can be dragged around and shaped and formed and then turned into something else. So it's kind of like rolling out your Play-Doh, and now you've got this, and then it becomes what it needs to be. Mesenchymal cells are, in embryos, we've talked about them a lot already. They're the cells that, in chick and mouse, they're mesodermal or mesendodermal cells that crawl in through the node and then crawl where they need to go. And then once they get where they need to go, they go from mesenchymal into epithelial cells, right? Then they start attaching to each other and forming tissues. So that's the MET transition. So cells can go in either direction, and they can go back and forth through those transitions as well. We saw that. Did we see that? No. Let's not go there. Okay. Okay. So hang on a second. Oh, you know what? I've got the wrong notes up. That's why I'm confused here. Here you are, you son of a person. Sorry for the delay, people. I know you're used to a more professional presentation than this. I apologize. Okay. So by the way, I didn't upload, I just remembered I didn't upload this PowerPoint or these notes yet. So I'll do that right after class. Okay, here we are again. So microtubules. Microtubules typically are cells that are not in mitosis. There's like one central area that they all come from. And this is really important for moving around vesicles in the cell. So if the cell needs membrane proteins, they'll be made in the rough ER, passed to the Golgi, and then passed out to the plasma membrane on these microtubules using these motors, kinesin and dynein, which are molecular motors. So if you've got a protein that needs to go to the plasma membrane, you put it in the membrane of a vesicle, and then the vesicle goes out to the plasma membrane and it fuses with the membrane, and boom, now you've got a protein out there. So that's a lot of what microtubules are being used for. Also, if we go back to chapter 4, we were talking about the cortical rotation of the egg after the sperm enters the xenopus egg. So, sperm enters and the cortex rotates. Well, the cortex is microfilaments, but that rotation is caused by microtubules. So you can take that home and tell your mother. Okay, the other thing microtubules are important for are in cilia, which we talked about in the last class. And cilia are kind of projections of the plasma membrane. Kind of the membrane just goes out. I was going to say invaginates. I guess it exvaginates. It goes out like this, and it's got microtubules in the center. And the molecular motors on those microtubules make the flagella flap in one direction. So it pushes a fluid in one direction. In the last class, we were talking about how the cilia in the bottom of this cup are forcing what is it? Is it BMP4? Oh my God, I've forgotten already. All right, don't look, don't look, don't look. Don't look, don't look. There it is. Shit. Nodal. See, I knew it was nodal. I knew it was nodal the whole time. So, so they're forcing nodal from one side to the other by moving the fluid over there. So that's what cilia are doing. And microtubules are in the cilia. And these motor maps, this dynein is in the cilia as well, and that's what causes the motion to happen. All right, but you all knew that. Microfilaments are a lot more flexible than microtubules, which tend to be stiffer. These are kind of rope-like. They're made of actin. And a number of different structures we see microfilaments in the cell. Let's see, we'll start here, I guess. These are kind of anti-parallel arrays of microfilaments. So they're opposite each other, and between them, there's a molecule of myosin holding them in place and also causing them to pass over each other to cause kind of a contraction of the cell. And that's how we'll get movement. We'll talk about that in a second, I guess. The other important one for us here is out here, the gel actin. Okay, so kind of like a mesh, a three-dimensional mesh of actin fibers that forms a kind of like a gel, and that's what the cortex is. This red here is actin. So it's the cortex all around the cell, and that's supporting the plasma membrane, giving the cell a shape, basically. And here's your mesh here. Okay, so what did I want to say about? Oh, so this is going to be used in cell crawling. So when those mesenchymal cells are crawling, this mesh part kind of polymerizes out, and then it puts down a focal adhesion and grabs extracellular matrix, and then it depolymerizes at this side. And then those anti-parallel stress fibers pull the cell forward into that area. And those depolymerized things from the gel get pushed through and they polymerize out in front. The integrins let go and they get passed forward and they grab further up, and that pulls the cell forward. So that's cell crawling. That's what a lot of these movements we see in the cell is all about. So you've got reaching forward, attaching, translocating into that new attachment, letting go to any attachments behind, and then reaching forward again. Of course, these aren't separate steps. It's all happening at the same time, but this is how those mesenchymal cells move around. Okay. So that's kind of broad general introduction to these things that we're going to be talking about next, which is, how do these cells move? How do they contract and change shape and everything? So let's go back and talk about adherens junctions again and how they cause shape and movement within a cell. So here's an experiment to show you how important these cadherins are. Now remember, cadherins only attach to a cell with the same cadherin in it, which means they're only going to attach to the same kind of cell. One type of cell will put out one type of cadherin. So in this experiment, they're going to take an early Xenopus embryo, okay, blastula. They're going to grab some mesendermal cells here and some ectodermal cells here, and they're going to treat them with collagenase or something to make them disperse into cells. Then they're going to mix all those cells up so they're totally mixed up and put them into cell culture. And what happens? They totally segregate themselves. So the mesendermal cells attached to mesendermal cells, ectodermal cells attached to ectodermal cells, and they're separate tissues again. Why is that? Well, if imagine you're a mesendermal cell and you're surrounded by ectodermal cells, you're not attaching to them because you don't have the right cadherins. Then you accidentally bump into a mesodermal cell. Oh, friend, good to meet you. And then you do this and you attach to each other. And another one bounces into you, and then you attach to that. Another one comes in, attaches to that. So just by randomly bumping into each other and only being able to attach to the same cell, they end up sorting themselves. And here the ectoderm is going to be on the inside. I'm not sure exactly why that is. Maybe, yeah, I don't know. I could make something up, but I'd rather not come out and do that now and make you more confused. So anyway, point is these adherent junctions cause tissues to adhere to each other. Why is that? And before we get into why that's important, I think I talked about this already because I was out of order, are EMT and MET. So this is an epithelial tissue here. So these are epithelial. These red spots are at adherens junctions. And when these adherens junctions let go, the cell then transitions into this mesenchymal form, and it can go in either direction, right? This is EMT, this is MET. And these are kind of loosely connected to each other. Sometimes they're not connected at all. When they are connected, they're connected, interestingly, by what do you call these guys? These are, gap junctions. So gap junctions are another type of cell-cell connection. The interesting thing about gap junctions is they're channels. So you've got a channel here and a channel here. They bump into each other, and then things can pass back and forth between the cytoplasm of the two different cells. So this is how they can kind of communicate with each other. Proteins can't pass between them, but small molecules and ions can go back and forth. So it makes them electrically connected, as well as other kind of signaling things can go back and forth between them. So that's your EMT and your MET, letting go of those adherens junctions, signaling the cadherens to let go. Now, let me back up a sec. We got 5 minutes left. Maybe I should let this go till next time. I think I kind of have these slides out of order. I'd rather talk about this than do this. So let's let that go till next time, because I mean, we're not in a hurry here, are we? We got all week. We got all week to talk about this. Who is Elmer Fudd? Who in the name of God is Elmer Fudd? That's what I got to know. Let's see your face, Elmer. Elmer, we're hunting wabbits. Elmer. Is cell biology cumulative? Yes, indeed it is. But it would be simple questions, though. There's Yusuf. Who's Elmer Fudd? I gotta know. I saw a rabbit in my backyard, Elmer.
Wait, sorry, what'd you say?
I'm asking who Elmer Fudd is.
Oh, I'm Elmer.
But there's an Elmer over here, though. Elmer Fudd. I'm asking you to unmute, Elmer. Who the hell are you?
Yeah, Elmer, show yourself.
Coward! Coward!
I guess we'll never know.
All right, I'm going to let you go. I will upload this lecture. You mind uploading this lecture right away? Yes, I will do this right away. And I will upload last Friday's lecture from a year ago. And I will upload the notes and PowerPoint from today, which isn't up there yet. See you guys on Wednesday.